Today, we are looking at cells and their organelles from chapter 3. The cell theory underlies everything that we know about cells and how they contribute to life. So there's 3 parts to the cell theory. You want to know these 3 parts. Cells are the building blocks of all plants and animals. All new cells come from the division of pre-existing cells. They don't just magically appear. And cells are the smallest living units that carry out all vital physiological functions. So they have nutrients that are turned into wastes, and then they also have some kind of respiration or photosynthesis that's going to help manage energy. Fun fact, cells maintain homeostasis individually. So each individual cell has to be in homeostasis, but then they coordinate to maintain the homeostasis at the level of the organism. So not only are they maintaining themselves, but they're also helping maintain the entire group. Cells differentiate and specialize, and this is governed by gene expression. So the fertilized ovum, so the fertilized egg, also known as the zygote, has the genetic potential to become any cell in the body. But as it divides, the cell is going to become its final functional form. So for example, in the zygote, we've got unspecialized cells, but we have more differentiated and specialized cells in the epithelial tissues, the connective tissues, the muscle tissues, and the neural tissues. I like to think of this as kids in a kindergarten. When they are young, they are not specialized. They may dream of being an astronaut and a bus driver and a fireman and a children's book author. But as they grow, they're going to go to school, they're going to become more specialized, and then they're going to have one specific function that they fill. And it will be harder to be both an astronaut and a firefighter and a children's book writer. Changing is harder once you're specialized in one particular area. There are 2 groups of organelles. So organelles are think of them as the little machinery in the cells that perform the functions. And they are simply intracellular structures with specific functions. So we have the cell here that is surrounded by the plasma membrane or the cell membrane. All of the fluid outside of the cell is known as the extracellular fluid. Once we get inside the cell, we have the cytoplasm. And the cytoplasm is dissolved into cytosol. So that is the actual surrounding material inside that cell, and then the organelles. And organelles are further subdivided into non-membranous organelles, which do not have a cell membrane or a membrane of any kind around them, and membranous organelles that have some kind of membrane around them, each individually. So we'll go through the organelles one at a time. I do suggest maybe some note cards to help you know. We want to know what's the function of each organelle specifically. So looking at a cell overall, getting ourselves oriented, we've got our nucleus, which is here in the center. You can see our little ribosomes. Those are the little dots and specks that we have on our rough endoplasmic reticulum. You have here your mitochondria. I think they look like beans with little squiggles in them. You have your cytoskeleton that's providing support and transport throughout the cell, and your plasma membrane here. We'll look at some other things as we continue going along as well. The cytosol is that liquid part within the cytoplasm. So remember, the cytoplasm is made up of the cytosol and the organelles. The cytosol is mostly water with dissolved salts and sugars in it. The organelles, they do very specific things that keep the cell alive and contribute to its function. We have the nucleus. That's the largest organelle in the cell, and it's filled with nucleoplasm. Similar to how the cell is filled with cytoplasm, the nucleus is filled with nucleoplasm, and it's surrounded by the nuclear envelope. So that's a double membrane, and it has little nuclear pores that allow things to go in and out, though there's a little bit of a guard system there because this is where the DNA and the control center is. You don't want everyone having control of the cell and how it's functioning. So the nucleus is going to control the metabolism of the cell. It stores the genetic information, and it's going to control protein synthesis. Fun fact, red blood cells do not have a nucleus. They're just being extra naughty, but it does play a role in how they function. So stay tuned for AP2. The nucleolus is located inside the nucleus. Some people will pronounce it nucleolus as well. Watch your spelling very carefully. Nucleolus is the little part inside of the nucleus. So make sure you know which one you're talking about. And the nucleolus is the site of ribosome production. So the ribosomes are what make proteins. The ribosomes are made in two parts. There's an alpha and a beta part. A finalized ribosome is too big to fit through these tiny nuclear pores. And so we're going to make it in two parts, send each part out separately, and then we will assemble it in the cytosol. Notice we've got our nucleoplasm that is all around inside our nucleus, and there is our nucleoli. Nucleoli is plural, a singular is a nucleolus that is going to produce our ribosomes. Vesicles are little membrane-bound sacs that will bud off the endoplasmic reticulum or the Golgi apparatus or even the cell membrane itself, and they're really important for transporting things around the cell. They provide just a little packaging to help things move. We have the garbage crew of the cell, so this is peroxisomes. Peroxisomes are a membranous organelle. They have pieces of the phospholipid membrane. So they're a little vesicle, but inside them they have enzymes. And so they're famous for breaking down hydrogen peroxide with the enzyme catalase. Hydrogen peroxide, peroxisomes, helps you remember a little bit what they do. Hydrogen peroxide is a reactive oxygen species and is dangerous to the structure of other parts of the cell. But peroxisomes will also break down fatty acids and help with the synthesis of cholesterol, bile, and myelin. So not only cleaning up, but also doing some synthesis. And we have lysosomes. These are membranous organelles. Again, they have that phospholipid membrane around them. They're a little vesicle that has digestive enzymes to break down organic compounds, old organelles, or pathogens for recycling. That's important to keep in mind. Tay-Sachs disease is an inherited recessive lysosomal storage disease where the lysosomes don't function like they should. This means that all of those old organic compounds, organelles, and pathogens will build up over time. And this occurs in young children, so starts at 6 months old. Usually we're seeing seizures by 12 months of age because we can't have all of these pathogens build up in the brain. And the cells are dying and triggering inflammation. These children will have visual and motor deficits and eventually will die at 4 to 5 years of age. So it just shows one small little enzyme can cause big problems in the whole organism. So we're looking here at the lysosomes and their functions. So an extracellular solid or fluid may come into the cells. Here we've got some lysosomal enzymes that are activated when they fuse together. This enzyme is going to break down this material. It's going to release the nutrients that are needed, and then whatever is waste, it's going to go ahead and let it go. Here you can see we've got some inactive enzymes. We've got an old organelle that's ready to be retired. The lysosome is going to bind to that organelle and digest it and absorb the nutrients as needed for recycling. And then here we've got a lysosome that hasn't needed to do anything. It's not been activated, and so it can break down, and it's not going to cause any issues because it's not been activated. And those lysosomes are coming off the Golgi apparatus. There are situations where those lysosomes have active enzymes where the membrane breaks down and it destroys the cell, and that's a process known as autolysis. And apoptosis happens in a similar way, but it is programmed. So when we talk about mitosis and cancer, we'll talk about this again, but you can have lysosomes that have inactive enzymes and won't do anything if they're breached. If they have active enzymes and are accidentally breached, the cell can start to digest and break down itself. Or if the cell or the nucleus has noticed some problem, they can plan for the lysosomes with their active enzymes to be released to kill the cell because the cell is unhealthy. So ribosomes are non-membranous organelles. Remember, we've got two units that are made in the nucleolus. They come out those nuclear pores, and then they're combined together. And ribosomes are responsible for protein synthesis. If you remember one thing from this lecture, remember ribosomes and protein synthesis. And protein synthesis means that each individual amino acid is assembled into a polypeptide. There are free ribosomes that are just floating around in the cytoplasm, and proteins that are made of these will enter the cytosol. But there are fixed ribosomes attached to the endoplasmic reticulum, specifically the rough endoplasmic reticulum, because these ribosomes are what make it rough. And these proteins will most likely be exported out of the cell for other purposes. Speaking of the endoplasmic reticulum, we have the rough and smooth versions. The rough endoplasmic reticulum, also called the RER, has ribosomes attached on the outside to make it rough. Once those proteins are made, they are modified and folded in the rough endoplasmic reticulum, and then they're packaged and shipped to the Golgi apparatus. And the rough endoplasmic reticulum is continuous with the nuclear envelope. It's kind of tubular, and its chambers are called cisternae. And we'll see that word again in some of our other organelles. Smooth endoplasmic reticulum, or SER, has no ribosomes, and it's involved in lipid synthesis. So specifically, we're thinking about phospholipids, cholesterol, sex hormones, glycerides, and those kinds of things. And it's going to synthesize and store glycogen in skeletal muscle and liver cells. So with our smooth endoplasmic reticulum, think about synthesis of lipids and glycogen. So we're looking at here our smooth endoplasmic reticulum that has no ribosomes attached, and it has these cisternae, or these cavities, that are tubular. And then here is our rough endoplasmic reticulum with our ribosomes attached, and they have these cisternae. They're a little bit more sheet-like looking. The Golgi apparatus, also known as the Golgi body, looks like a stack of pita breads or a stack of pancakes. And we get these flattened membranes that are called cisternae. So again, this space between membranes, we're seeing the repetition of this word. And they store, they alter, they package, and they ship all sorts of products, whether that's a protein, carbohydrates, or a lipid. I kind of think of them as the Amazon fulfillment center. So they'll get the materials, they're going to get it packaged, and then ship it out where it needs to go. It's also responsible for renewal of the cell membrane. So little pieces of vesicles and the Golgi body will go and actually replace the cell membrane that might be damaged or might need more. Here we see that we're getting some secretion of product into this extracellular fluid, and that happens via exocytosis. And then the Golgi is also where those lysosomes come from that are going to hang out in the cytosol. So the cis and the trans sides are not political in biology, but they are the receiving side for these vesicles, and then the transport side where the Golgi are going to send things out. So things usually go in the cis space and out the trans space. The mitochondrion, people always know them as the powerhouse of the cell. Please, please, please don't ever tell me that. If you write that on a test, on a quiz, on anything, it will be wrong, because I can guarantee you say powerhouse of the cell, but you don't necessarily know that that's the place for cellular respiration and energy production. So you can remember it powerhouse of the cell, but don't write it down. Tell me what it really does. The mitochondrion has a double membrane, so the inside membrane is folded to increase the surface area for proteins and enzymes, and here we have little folds called cristae, and this is where ATP production occurs. And you get the most efficient ATP production if oxygen is present. Oxygen can be the final electron acceptor of the electron transport chain, and it permits oxidative phosphorylation. So we're going to get the most ATP out of each organic molecule that is possible. So quick introduction / review of aerobic respiration. There are 3 main stages, so you might want to know where the stage takes place, and kind of in general what it's doing. So the first stage is glycolysis. This occurs in the cytoplasm, in the cytosol, and we're going to split glucose in half into pyruvate. Pyruvate is going to go into the citric acid cycle, and it's going to be broken down more. And then finally, we're going to get to the electron transport chain and ATP synthase, and that's going to use the electrons that we've gotten from the first 2 steps. So from glycolysis and the citric acid cycle, they're going to go down a chain towards oxygen. Remember from chapter 2, oxygen has that high electronegativity and attracts those electrons. And as it goes down the chain, the energy is used to add a phosphate to ADP, making ATP, which is that energy currency in the cell. So I'd know about the 3 stages. I'd know that the electron transport chain is where most of our ATP is made. The citric acid cycle happens in the mitochondrial matrix, and then the electron transport chain happens in the mitochondrial membrane. So keeping track of where those occur as well. Once again, we're seeing the formula for cellular respiration. So we have glucose and oxygen as our reactants that produce carbon dioxide, water, and about 38 ATP. You can see the double membraned mitochondria. In stage 1, we have glycolysis here in the cytoplasm. It's going to move into stage 2, which is the Krebs cycle, also known as the citric acid cycle, and then to stage 3, where we have the electron transport chain and ATP synthase. That is where most of our ATP is going to be produced. The cytoskeleton is the cell skeleton, and it provides strength and flexibility. So there are several different molecules that we see under the heading of cytoskeleton. We have microfilaments, which are particularly organized in skeletal and cardiac muscle cells. We have thick filaments that are found in skeletal and cardiac muscle cells, as well as intermediate filaments. Microtubules are in most cells, and they will actually radiate away from the centrosome to provide strength and move organelles. Centrioles are made of cytoskeleton particles, so they're made of microtubules, and they're going to help move the chromosomes during cell division. We have cilia and flagella, which extend out of the plasma membrane. Flagella is the little tail that moves the sperm, and the cilia are these projections that are finger-like, and they can wave back and forth and move things along them, such as debris that might be in the respiratory tract that could cause issues. It's going to try and move that debris so it doesn't go deeper in the lungs, but so that maybe you're able to cough it up. And microvilli are another example of part of the cytoskeleton. These are the extensions of the plasma membrane around specific cytoskeleton parts, so around the microfilaments, to increase the surface area so we have more area to absorb things, and this occurs in the intestines. Instead of having a flat surface, now we have all of these extensions where there's more space for absorption to happen and be more efficient. We are going to stop at protein synthesis, which looks at how organelles work together, and we will continue this in class.